So today, I'm going to talk to you about our quantitative methodology using fluorescence lifetime imaging microscopy. And we're going to talk about our method to image the temperature in cells by using FLIM. And then also I'm going to talk about some of the application of a method to understand the novel thermal signaling, which is a mechanism of cell functions. So first I'd like to talk about my motivation of our work. So this is an electron micrograph of a living cell, a single cell. You can see some nucleus and some other important organelles. And you can see there's many molecules in highly dense and packed and very crowded environment. So what is like the local environment field in this space? So my motivation is to understand the dynamics of this environment itself and the molecular behavior. どうもありがとうございます。これでいかがでしょうか。So my motivation for work is understanding the dynamics of local environments field inside of a single living cells. So this electron micrograph clearly shows that the inside of a cell is not equal to the pure solution, which is mainly presumed in the biochemistry. So here I'd like to talk about the intracellular nanoscale space, which I would say like the size of sub-micrometer scale, and there exists unique physical-chemical environment, such as temperature, pressure, and viscosity or something like that. And this microenvironment define the state and dynamics of molecules in this area. So that therefore, this environmental fields should have a huge impact on cell functions. And I believe that understanding this intracellular microenvironment leads to uncover novel concepts in biology and of course in nanotechnology assisted future medicine. How do we investigate this environmental field? Of course, we require quantitative description of behavior by molecule in this environment and also environment parameters itself. On the other hand, conventional cellular imaging has a highly limited only to the qualitative understanding of the distribution of the specific molecule. To tackle this issue, we use single molecule detection imaging. I will refer SMD imaging. So back in the 1990s, single molecule imaging technique by using total internal reflecting fluorescence microscopy TOF has highly flourished based on evanescent field, which allows high signal to noise ratio. And also we can visualize single proteins and many motor planes was being investigated using this single molecule imaging technique. And this single molecule imaging technique can be applied to cellular imaging by using single molecule detection technique as a single SMD imaging techniques. So I would list some of the SMD imaging techniques, such as fluorescence lifetime imaging microscopy FLIM, or stochastic optical reconstruction microscopy STORM, fluorescence correlation spectroscopy FCS, and fluorescence recovery after photobleaching FRAP, or single particle tracking SPT. So I utilize all of these techniques to understand the behavior of the molecule or the environmental parameter inside of living cells. The feature of this SMD imaging technique is highly quantitative and high sensitivity and high accuracy. And also, I'm happy about highly multimodal. So in a couple of slides, I'm going to talk about the SMD imaging techniques. The most famous and the fundamental technique for SMD imaging is nanoscale localization with single fluorophore with fitting. This was conducted in 2003, and so the SMD imaging is based on two simple procedures. The first one is acquisition of numerous photons. So we can acquire a lot of photons over time, and in this case, using the very bright dye and accumulate the lots of photons over time, and when you can described as like a distribution of the photon in space. And then the second factor is approximation with the theoretical function. So in this case, that's using the Gaussian fitting, which is pretty simple, and then we can determine the center of the position of this dye. And the full width is equal to spatial resolution with only 1.3 nanometer, and which is much higher than the conventional diffraction limited spatial resolution. So again, I'm going to stress on that this SMD imaging, first we acquire lots of photons, and then using the theoretical functions and fitting. So because we use fitting, approximation, we can obtain other validative data, such as errors or the detection limit. That's a very important aspect of this MD imaging. And this nanoscale localization techniques was allowed for the stochastic optical reconstruction microscopy done later, and it is pretty famous. I don't have to talk about it right here. And FLIM is also another SMD imaging. So this is a pretty simple cartoon for the fluorescence. So when you excite the fluorophore with a certain energy, such as light, and the fluorescence molecule goes up to the excited state, and then gradually relax to the ground state. And when you use a pulse laser, so only the excite fraction of the fluorophore, and then stop exciting, so some of the molecule relax to the ground state by emitting photon. And when you recorded this photon decay as a function of time, we can draw the fluorescence decay curve. And this is called time-correlated single photon counting, TCSPC. This is one of the most accurate fluorescence lifetime detection mechanism approach. When we do apply this technique into the fluorescence imaging, especially using confocal fluorescence microscopy, so we do this. So we pixelate some of the sample, and we irradiate each pixel by using pulse laser by scanning, and then obtain each fluorescence decay curve from the all pixels, and then fitting with approximation using exponential decay. In our case, we have two distinct components of fluorescence decay. So we use the two double exponential decay. And then we obtained each average fluorescence lifetime, and then we constructed fluorescence lifetime image. So again, in the FLIM methodology, we photon counting lots of photon, and then followed by approximation. So by using approximation, we can evaluate the accuracy or the error of this methodology. Here I'd like to summarize some of the advantages and the limitations of the FLIM. So usual fluorescence imaging is fluorescence intensity imaging, which is, of course, we have an advantage such as highly sensitive and high spatial resolution. And of course, when you look at using very fast scanning or high-speed camera, we can go into the high temporal resolution. And the limitation is, so when you just look at the fluorescence intensity, which depend on the chromophore concentration and light source strength. So of course, we have to, when you obtain very quantitative data, you have to normalize the intensity or using the ratio. On the other hand, when you're using the fluorescence lifetime imaging, which does not depend on the chromophore concentration, we don't have to bother from this artifact. So the most high advantage of this methodology is chromophore independent imaging. And furthermore, for me, what's more important is the fluorescence lifetime highly depend on physical chemical property. So the motivation of my work is understand the image of temperature in cells, which is one of the most important environmental parameters. So I don't want data affected by other physical chemical properties. So when we obtain the fluorescence lifetime, we also evaluate the other contribution of other parameters. The limitation of a fluorescence lifetime is unfortunately highly expensive, and also resolution will be low because we have to require lots of photons. So we have to compromise this part. And when you compare with the fluorescence lifetime imaging and then fluorescence ratio imaging, I would suggest that fluorescence lifetime imaging is much more precise because ratio imaging is sometimes somehow independent of chromophore concentration, but at the same time, when you go into extreme concentration, the ratio will be affected. Maybe you know, some of you know. So I would say, yes, and of course in ratio imaging, you have to use 2 dyes. So fluorescence lifetimes is much better than ratio imaging. So again, the feature of FM imaging is highly informative and the possibility of validation. So what informative means, so usually we can obtain the multimodal such as the precise position of the fluorophore or the diffusion state, or that maybe we can track the trajectory to understand the state of the molecules. And speaking of the validation, so basically, the fluorescence imaging lacks in the validation so far, but in SMD imaging, we can go into the quantitative, and also we can discuss the precision, accuracy, or limit of detection, which is very highly advantageous when you think of the analytical chemical perspective. However, we have to compromise the spatial temporal resolution for informative outcome, and vice versa, because we have only limited photon budget. When you're given a certain fluorophore, such as a GFP or even any other dye, has a only limited number of the photons that can emit from the one single dye. So we can, if you wanted to go into the higher spatial resolution, we have to compromise the information. Or if we're going to get the very important resolution, we have to compromise the information. So to be honest, I would compromise resolution when I image in the temperature in cells, because temperature is very interested me. So from here, I'd like to talk about our application of FLIM into our research. So my research is about the temperature, related life activities, which is called thermobiology. I have to apologize, some of the data I'm going to show you from now is overlapping the one I presented yesterday in the symposium thermobiology. So this is the background of the thermobiology. So now not to mention temperature is highly important in various level of life activities for molecule to cell, tissue, and individual. And some important mechanism is already known for contributing this temperature-dependent life activities. And of course, I'm interested in intracellular space, like I mentioned earlier. And my research target is try to investigate intracellular local temperature in this very highly crowded environment, which I believe this contribute to cell functions. So the methodology of imaging the temperature in cells highly limited. The conventional methodology to imaging temperature, such as calorimetry or thermocouple thermography, cannot apply to a single cell level because of the size and sensitivity of the methodology. On the other hand, molecular thermometer, which can function at the molecular level, can allow us to measure the temperature even in a single cell. So this is the cartoon of the diagram, cartoon of the fluorescence molecular thermometer. So when you're given the fluorescence property is drastically changed according to the temperature, we can fluorescence image this fluorescence, then we can determine the temperature. So far, some various kinds of thermometer has developed from using small compounds, synthetic polymers, GFP, and some inorganic nanomaterials. So among those candidates, we use fluorescence polymeric thermometer, FPT, because this is one of the most sensitive thermometer applicable for the cells. So this is a chemical structure of FPT, and which comprises of the polyacrylamide, which has a 20,000 kilodalton molecular weight. And this comprises of the amido bond and alkyl chain. And this hydrophobic and the hydrophilic moiety competing at the lower temperature, but when you heat up the solution, the hydrogen bonding becomes relatively weaker, resulting in the hydrophobic interaction of this alkyl chain to assume the shrink structure of the sequence. And we also introduced some of the environmental sensitive fluorophore benzofluorescence. So this is highly quenched at a lower temperature because of water molecule nearby. But the higher temperature, as I said, just shrinks of the main sequence, running out of the water molecule and fluorescence highly recovered. So this is a fluorescence response of just a combination of the acrylamide and benzofluorescence. So as you can see, the temperature-dependent fluorescence jump can be seen here. This is based on the phase transition of the acrylamide. We have to apply a lot of fluorophore and also hydrophilic unit to make this FPT stable even in a living cell. So the response is a little bit hampered, but still we have obtained a 5 times enhancement of fluorescence intensity. The earlier study, we have investigated sensitivity of a thermometer, which has a highly, we can detect only 0.1 degree difference, and also functional independency of the thermometer. Other chemical parameters such as pH or ionic strength, the concentration of a molecule and by viscosity do not affect the function of the thermometer. So back to the detection of the fluorescence. Fluorescence lifetime, like I said, an index for accurate detection, because usually fluorescence intensity depends on the quantum field and concentration, but fluorescence lifetime does not depend on the probe concentration. But the fluorescence of a thermometer highly dependent on the temperature. So we obtained the performed the TCSPC over 50 to measure the temperature in cells. So this is a temperature-dependent fluorescence decay curve we obtained from using the cell extract. So this is the result in the solution. So you can see the higher temperature fluorescence decay curve becomes longer than when the lower temperature. And you can see the 2 fractions of the components of the fluorescence decay. This is coming from the 2 states of a dye. So we have various unique fluorophore and a lot of hetero atoms which can form the hydrogen bond. So either hydrogen bonded fractions or non-hydrogen bonded fraction result in 2 fractions. But anyway, the fractions of is not really different between the temperature. So we can just compare the average fluorescence lifetime given in each temperature. So this is a calibration of the fluorescence lifetime as a function of the temperature. So we can measure the temperature just measured by acquiring fluorescence lifetime in each pixel in the cells. And again, the temperature resolution from this calibration curve gives us 0.2 degrees. So how do we do the image of temperature using the FPT? So first we introduce FPT into the living cells by either microinjection or using cell permeation. By changing the chemical structure a little bit to the cationic structure, this molecule goes into the cell membrane immediately by co-culturing. And then we measured the fluorescence lifetime using TCSPC. So now I would like to show you the temperature distribution in the single cells. So this is a confocal fluorescence image of the thermometer in cells. As you can see, some of the locations becomes brighter, so maybe it comes from the higher temperature or just concentrated fluorophore. By acquiring the fluorescence lifetime image, in this case we accumulate 60 seconds of the photons and also 64 by 64 pixels, which is a really low spatial resolution. We have to compromise because we require lots of photons. As you can see, some of the area, the nucleus is seems yellow to orange, whereas the cytoplasm becomes green to blue, suggesting that the nucleus is supposed to be warmer. So detailed imaging investigations discovered that some important organella is warmer than another, such as nucleus is warmer than the cytoplasm by almost 1 degrees, and which is dependent on the cell cycle. And also, when you zoom into the area of the cell, some of the mitochondria show higher temperature, even in a steady state. And on the centrosome also show a warmer temperature. The mechanism and the significance of this temperature distribution in the cell is still under investigation. Some of them we have some clues, but most of them is still in the mystery. So from now, I'm going to show you some of the results of the imaging of the intracellular temperature using various kind of samples. So in this slide, I'm going to show you the temperature distribution in the mouse brain slice. By using the cell-permeable FPT, we can easily stain the cell in the mouse brain by FPT. And by using the FLIM, we can visualize the temperature. So this is like a control experiment. When you change in the temperature of the medium, the fluorescence lifetime becomes longer. So this can be a calibration curve determining the temperature in the cell, intracellular temperature in the brain slice. And when you go into the deep area of the slice, we can very interesting temperature distribution, such as a very heterogeneous temperature distribution in the cortex layer 1 or hippocampus neuron, with the dendritic structure has shown the different temperature. And also cell body is also some heterogeneous temperature distribution, even in a steady state. And furthermore, we introduced a thermometer into the C. elegans to understand the temperature distribution in the individual level. By introducing a thermometer into the gonad, and then we just wait for this probe goes distribute evenly into throughout the oocyte. So this is a fluorescence lifetime image of each temperature. You can see that the temperature increases, the fluorescence light of the thermometer becomes longer, suggesting that the thermometer can function even in the C. elegans. As you can see, some interesting temperature distribution in the nucleus of gonad, suggesting the higher activity of the nucleus in the very primitive oocyte. And so personally, we are interested in the correlation of the RNA granule and the temperature. We just visualize both the mRNA and also temperature. And so this is the embryo. So you can see some of the germ cell located in the posterior of the embryo is showing higher temperature. And when you look at the oocyte, so this is the black area, the nucleus, and so this white spot is the P granule, which is responsible for translation regulation. So we can see some of the warmer temperature is seen near here. So we just speculate maybe the higher temperature and RNA granule formations is related. And so this is some of our ongoing investigations. So anyway, yes, our thermometer and FLIM can allow us to image even in the individual level. And when you look at the neurons, this is a dispersed culture cell, culture neuron. And when you look at some small structure, so we believe that this is spine and confirmed by immunostaining. And some structure showing the warmer temperature, and some of them is very cold temperature. And so I like to highlight the very dynamic temperature in this structure. So this is also another ongoing project. So yes, so far we have shown you that I have convinced you about the methodology to image the temperature by combination of FPT and FLIM. So what is the impact of a temperature variation in single cells? So the temperature change observed in cells is 1 to 2 Kelvin in a single cell, which is highly unique compared to the water. So this, I don't want to talk about this detail, but when you just calculate the possible temperature change using the heat equation equation, the temperature change supposed to be happening is 1 to the minus 5 Kelvin, is with significantly smaller than we have observed in cells. This is coming from the unique chemical environment, and we're going to talk to you about the mechanism later, but the thermal diffusion is highly restricted in the cells. And so because of the drastic change of the chemical environment in the cell with respect to temperature, so the heat has a huge impact in the cells. So therefore, we hypothesize the local heat must have a very important physiological function in cell, so which we call intracellular thermal signaling. So in a couple of slides, next couple of slides, I'm going to talk about our investigation about thermal signaling. So we investigate the thermal signaling to show that the hypothesis that intracellular temperature variations in the local area contribute to cell functions by using measurement of the local temperature and also manipulate the local temperature by using light. So this is one of the approach that we manipulate the temperature, heat in the local area of the living single cells. By using 1480 nanometer infrared laser focused on the surface of the glass, we can stimulate the water directly to warm up. So this is the result in the solution. You can see we can change the laser power. We can heat the solution in a quantitative manner. So when you apply this methodology into the cell, of course we want to do this in a quantitative manner. So FLIM was indispensable because we have to calibrate the temperature change when we apply the artificial heat source in the cell. So even in a COS7 cell, we can heat it up the local area. I would say the 2 micrometer area in the cells in the laser strength dependent manner. When you look at the temperature change in the solution in the cell in the same laser power, the temperature increase in the cell is higher than in a solution. This is interesting because this suggesting that heat conduction is limited even in the living cells. But anyway, we can heat the local area of the cell using this. And the phenomena we apply this technique is stress granule formation. And stress granule is the transient organella which is responsible for translation regulation. And so this is a non-membrane structure containing housekeeping mRNA, 40S ribosome, translation factors, and so on. And so this is the fluorescence image of stress granule formation. And so we can see that formation is pretty quick. And when you remove the stress, it can go back to the diffusing state. So the mechanism of the stress granule formation has been suggested that the liquid phase separation of the SG components, but no one knows the mechanism directly initiating stage formation. By looking at some properties of there is no membrane or rapid assemble, disassemble, we speculate the temperature, intracellular temperature contribution in this initiation mechanism. So first we perform the image the temperature when stress granule formation. So one of the most important, famous SG inducer is oxidative stress using arsenide. And when you apply the arsenide, which is like a toxic to the mitochondrial proteins, we observe that significant temperature change. So this is 30 minutes after the induction, so the temperature change was 1.4 degrees. And interestingly, this temperature change happening at exactly the same time course of the stress granule formation. And another stress causing SG is ER stress using Tufts gouging. And in this case, a similar result was obtained by putting Tufts gouging. We observed 1.4 degree increase in the cell. So temperature increase and somehow related to stress granule formation. But we don't know yet whether temperature is the driving force of stress granule formation or just the outcome of this phenomena. To prove this, we provoke the heat thermogenesis in cells. So like I said, mitochondria is known to produce the heat in cells. And so FCCP is one of the strongest uncoupler, which result in the leakage of the proton and temperature of the cell increase. And the temperature increase reached to 1.3 degrees over 30 minutes. And when you observe this mRNA distribution, you can see the transient, beautiful SG structure formation was observed upon mitochondrial stimulation. So the heat production from mitochondria can provoke stress granule formation. And furthermore, we applied our artificial heat cells to provoke stress granule to quantitatively understand the heat-assisted SG formation. So this is an artificial temperature gradient formed with infrared laser irradiation. And when you irradiate this area of the cell, we can observe the immediate stress granule formation. And this is not happening to the adjacent cell. So we're suggesting that we can manipulate the stress granule formation in the single cell level. And this formation was dependent on the strength of the laser power and the duration of the irradiation, suggesting that the temperature change dependent SG formation. And so also we quantified the temperature change required for stress granule formation by applying the same amount of heat to the temperature of the medium of the cell. As can be easily imagined, the higher temperature resulted in the stress granule formation. But at this point, we can't distinguish between delta T or absolute temperature, which is important. By applying quantifying the temperature difference, which is required for stress granule formation, told us that the temperature difference required for SG formation was absolute temperature of 42 to 43 degrees in the local area of the cell. So in this case, I want to make a stress that the 42, 43 degrees is only 1 or 2 micrometer in a single cell, so which is distinct from the classic heat shock response. And together with other investigation is a molecular mechanism. This is a hypothesis of stress granule formation dependent on the local thermogenesis. So when the cells are exposed to the stress, the cell produce heat from organelle, such as mitochondria or ER, and this promotes the denaturation of the translated mRNA to making the naked mRNA, and this diffuse away from the heat source to the colder area of the cell and make a granule. And famous biochemical signaling or phosphorylation of IF2 alpha is also contribute this phenomena, but the heat is more dominant, and this can prevent the back reaction to the ribosome rebinding to the mRNA. So this is one of a approach to understand the thermal signaling, which I'm very happy because not only the heating technique, but the first temperature imaging was indispensable in this project. And another example, I want to show you another example of thermal signaling, so very briefly, which is brain edema and contribution of TRPV4. So brain edema is characterized by the swelling of the brain, and which is provoked by ischemia condition. And TRPV4 was suspected to involve in this phenomena. And we first investigate the contribution of the TRPV4 into the brain edema. And so this clearly shows that the ischemic condition by applying oxygen glucose deprivation resulted in the swelling of the brain size, and this does not happen in TRPV4 knockout mice, suggesting that TRPV4 is contribute to edema. But where does the heat come from? Because TRPV4 is known to be activated by heat. We suspected that contribution of the endogenous heat production in the neuronal cells. And we use our image of the imaging the cellular temperature in the brain slice. When you apply the ischemic condition, you can see the significant temperature increase in the cell in the brain slice. They calculate the temperature difference with the 2.1 degrees, which is significantly higher and enough for activating the TRPV4. Because TRPV4 is opening at the temperature of like 38 or 40, and when you think of the body temperature, this temperature is enough for the activating TRPV4. So ischemic condition result in intracellular heat production, and also heat production coming from neural activity by investigating the temperature in a glutamate treated cell. So you can see when you apply the glutamate, the temperature significantly increased by a couple degrees, and this could be inhibited by applying glutamate receptor agonist. So ischemia result in a neural activity, and this provoked the heat. So this is this is a suggested model of hyperthermia-induced edema. So we know that the ischemia result in stroke by water influx into the cell after the TRPV4 opening. And we identified intracellular temperature increase is the mechanism to open TRPV4, and this coming from neural activity. We haven't determined the precise mechanism of heat production, but we speculate the contribution, transcription, and inflammation. So I would like to summarize some of the outcomes of the intracellular thermal signaling. So local temperature change in living cell does facilitate the function. And the feature of the thermal signaling is pretty rapid, happening at the second to minute order, and also characterized as high efficiency of energy utilization. And also very interesting phenomena of the this happens only specific temperature window, which is, so that this response is non-linear response to the temperature. So now we investigate the mechanism of thermal signaling by either molecular mechanism, by looking at like mRNA, tracking the fusion mRNA using SMD imaging when in the timing of the thermal signaling, but also in a physical perspective. Thermal propagation in the cell has something to do with thermal signaling because the molecule has to utilize the heat. So especially heat dissipation from a thermogenic organelle is important. So the rest of this talk, I'm going to talk about the mitochondrial thermogenesis and our attempt to measure this temperature thermogenesis in the cell. So like I said, FCCP was one of the strongest uncoupler reagent, which leaks the proton into the matrix of the mitochondria, resulting in the huge heat. When you apply the higher concentration FCCP, we can record 1 degree increase on average in the cell. And when you culture the cell at the lower glucose concentration, which sort of starves the energy, the activity becomes pretty low. In the case the temperature increase from the mitochondria is highly suppressed. So this temperature change can be used for the calculation of the energy conversion to the temperature. And I don't want to talk about it, but almost half of the energy converted into the temperature change. And again, back in our stress granule formation, we applied FCCP and the temperature increase and the stress granule formation. So earlier, we tried to image the temperature near mitochondria in a steady state. So this indicates the location of the mitochondria, visualized by Microscopy. And when you look at the temperature distribution near the mitochondria, some of the area shows higher temperature. But this not happen with all mitochondria. Just very specific mitochondria. We didn't know the reason, we don't know the significance, but very specific areas is warmer. And when you stimulate the mitochondria by CCP, so this is like a time-lapse image, only the 2 image of time-lapse, but at 30 minutes, so the temperature of the overall area increase. So maybe the temperature increase coming from the location of mitochondria and dissipate to the cytoplasm, but we couldn't detect this transition because so in the fluorescence lifetime imaging technique, we cannot apply the multiple procedure of imaging because our limitation to our dye. So unfortunately, I have to confess that our dye is a little dim, so we have to consider the photo bleaching. So that the maximum time we can time lapse was 2 so far. So here, I'd like to introduce new Nobel FALCON FLIM. So this is a pretty novel system from Leica. And so this is not my data, but I just get from the website of Leica Microsystems. And so Nobel FALCON is fast lifetime contrast, whatever. So this is very prominent methodology in FLIM because, so what is distinct from the conventional FLIM as to the photon counting efficiency is usually highly sensitive. So because this has a sensitive detector, which is suitable for SMD imaging, and the second factor is digital noise removal, so minimizing part-up effect. I don't going to talk about the part-up effect, which is a very fundamental issue when you're doing the FLIM procedure, but this is sort of problematic when you do multiple imaging or like fast acquisition of the FLIM, but we don't have to worry about that. And also efficient scanning. So all these 3 factors contribute to the better detection efficiency. So now we don't have to worry about the photon budget to which we prioritize resolution or information. Because using FALCON, we can obtain high resolution and high information imaging. So this is the simple comparison of a method of result of the FLIM. So this is one of the results that I showed you earlier. So this is a conventional FLIM image. This is accumulation of 60 seconds, like I said. And so this is a single cell. And when you look at the local area, we can still tell that the nucleus is warmer, but the resolution is really low, and the temperature distribution is very blurred. When you're using the FALCON FLIM, accumulation of this required for this image was less than 1 second. I would say 100 milliseconds. And you can see the very precise distribution of the temperature. I have to confess that some of the unwanted feature of a thermometer distribution, such as some black darker area, which probably FPT does not diffuse into this area. And so seeing very precise is not always happy, but this is very much better than compared to this. So when you look at the local area, we can obtain the very clear temperature gradient formed in the between the nucleus and the cytoplasm. So we are very happy about to see this because FALCON FLIM gives us much more precise temperature distribution in a cell. So back to the heat propagation in a cell, one of the most wanted result was imaging the heat propagation in a cell when you stimulate the mitochondria. So I'm going to show you the latest outcome of our investigation, the imaging thermal propagation in a cell. So I'm going to explain the experimental condition. So we monitor the temperature over 30 minutes, and we check for the bleaching was minimal, even though we perform a couple hundred times of imaging, because the FALCON is highly sensitive, so we can reduce the excitation light source. And we check the fluorescence decay curve in every image, in which was excellent. So the experimental procedure is pretty simple. You just put in the SCCP into the medium of the cell and just time-lapse imaging of the temperature using fluorescence lifetime imaging microscopy. I want to make a stress that the fluorescence type of imaging is not usually used for time-lapse imaging, but FALCON is highly sensitive and minimum photopolishing. We can perform quite a video using FLIM. So this is the starting temperature distribution. This is a nucleus, and some of the darker areas are mitochondria. And so what I expected was, so maybe some spot-like burst-like temperature increase near here. So this is like a movie of the temperature over 30 minutes. So as you can see, the temperature is gradually increased evenly throughout the area of the cell. So this area, darker area, is the mitochondria, but does not necessarily producing heat. The temperature is gradually increased. That was a huge surprise for us because temperature is supposed to be heat supposed to be produced in the local area. So it might be a little bit of confusing because I am also confused about this data, but the results show that the temperature gradually increased, not in a temperature increase in local area. And this means heat propagate evenly throughout the cell. And so this is potentially contributed by the heat conductor. And I'm not going to mention about the mechanism, but also we discovered that the microtubule is indispensable for the heat propagation in the mitochondria thermogenesis. So maybe heat does not release upon stimulating mitochondria immediately after stimulation, but that maybe heat conduct to the microtubule, some other molecule, and then gradually propagate through the cell. So if this is true, this is a very important discovery in this area. So yes, so I'm happy to summarize my talk today. By using FPT and FLIM, we can measure the temperature distribution in the cell, and also using the heating technique, we discover thermal signaling in the living cell, as well as unique characteristic of thermal signaling, including heat propagation in the cell. In the future, I'd like to do more biological medical significance of a finding, and also the physical and biological mechanism of this thermal signaling. And also, I would like to thank my collaborators, including Benishi and Dr. Koyama, and some people from the Excels and Leica people. Thank you very much for your kind attention.